Hello, my name is Stephan Züchner. I'm a professor for human genetics and neurology at the University of Miami. Unfortunately, I cannot participate in the SPF meeting this year, but we agreed that maybe I can pre-record my slides and it will be interesting for you to see those. So let me share my slides with you. Okay. So I want to talk about the opportunities that we have in HSP genomic research and what this also can mean for therapy. And especially in the context of developing a new consortium in North America for centers of excellence for research in HSP. We call this Hsp-Cern. And my colleagues here, Caig, Blackstone, Rebecca Schüle from Germany, and also Darius, Ebrahimi-Fakhari, who's there at your meeting, sort of join me in these overarching plans. And here I have a bit of a background of these four sort of founding members. Rebecca is a professor of neurology in Heidelberg. She's the leading really scientist for HSP in Europe. And I think she's very well known to many of you. Dr. Ebrahimi-Fakhari is an associate professor at Boston Children's Hospital and has really already made a name for himself by conducting natural history studies and clinical outcome studies in HSP. And of course, Caig Blackstone doesn't need introduction. He's a professor of neurology at Harvard University, and he really is interested in exploring the molecular mechanisms of HSP. So as we have shared with your board, there is a unique opportunity to develop these centers of excellence. And I'm sure you will talk about your meeting in more detail about these plans. And this is really by learning also from other diseases and disease fields of rare diseases and what they have done to progress towards therapeutic solutions. So we think that such a network of academic scientists, we can reach more patients to participate in research studies. By working together, we can certainly find synergies. There are, once we start such a network, there will be different opportunities in terms of funding. We will have a much stronger case to make to get significant funding, for instance, from the NIH, the National Institutes of Health. And really the key goal of such a network would be to obtain clinical data from patients, create genetic data and house them and share those data, and also have a registry for patients available. So with this, I'm going to start talking about a little bit about the state of genetics in rare diseases, especially. As you surely know, genetics has made tremendous progress in the recent decades. And much of this has been driven really by technological development. And technological development leads also to reduced cost. So today we can sequence a human's genome for about $500, and the prices are still sinking. Only last year, which is interesting, the sort of final version of the human genome has been put together by scientists, and this has been published in the Journal of Science. We call this the telomere-to-telomere genome. And here I have three names of companies: Illumina, Nanopore, PacBio. They will sort of represent the leading edge of technology in our field. And while all these successes have been remarkable, there's actually a new technology now available called long-read whole genome sequencing, which we think will again change the picture and will allow for many more discoveries. So here is an overview of all the many genes that can cause SPG or HSP. As you know, there are many different genes. It's very heterogeneous. And yet with all this ability and all these discoveries, there's still about a third of all patients, and probably this number is even quite a bit higher. With tremendous efforts, we cannot identify the underlying cause, genetic cause, in a particular patient or family. So we really want to get to a much higher rate of probably over 90% of diagnostic rate. And what we have learned by discovering all these many genes, and doctors have been looking carefully at these patients, they found that really HSP doesn't live in a silo. It is connected with other disorders that affect nerves in your spinal cord, but also nerves sometimes that run in your arms and legs. And there are these connections with related disorders, and we call this a spectrum, which really ranges from diseases that affect peripheral nerves, then also central nerve tracts, like in classic HSP, but also diseases that are called ataxias, that are often or that sometimes can be seen in combination with HSP. So we really want to take this all into account when we talk about HSP. To tackle the task of diagnosing more patients, one has to really understand the genetics of HSP patients. So by collecting data sets, genetic data sets, we can learn about new genes. We can also learn about existing HSP genes. And here is a quick overview of our database, where we currently have over 2,000 HSP genomic data sets available. And in the past 5 years, this is a typical time frame for an NIH grant. We added 1,300 HSP genomic data sets. So there's good progress, and with a unified consortium, this will only get better. These data are actually available to researchers in a tool that we call GenePair, and anyone can really go there and look up variants in a de-identified fashion. Look up variants in HSP genes and in any gene really in a human genome. And so scientists can see if there are interesting signals, and then they can communicate with each other and find each other. We also increasingly want to make existing data available at a high granularity, so that researchers have to register first to ensure safety. Data is always de-identified, so patients could never be identified from such data sets. But at the same time, we have to give researchers access to really discover new things, and possibly on an international scale, really. So here is our current sort of state, where we have about 18,000 genomic data sets from those related disorders that we have already collected. This database is certified really to be used under, for instance, the European data privacy requirements. And just very recently, we have now the ability to share data freely with all registered users. And for instance, I myself, and other collaborators like Dr. Shai from Iowa, we have now made our data fully available without any grace period. And we are trying to convince others to do the same. This will really expedite science. And as you can see on the right side, these efforts, these databases are supported by other organizations, such as, not unlike SPF. And that is very important. Now, here's a typical example of a gene discovery, UBAP1, that was made a few years ago. And because of this international database, we can immediately find these rare families in different countries. And that leads to a much stronger case for a new gene, and also sort of expedites follow-up studies. Having such data available attracts, of course, also scientists who develop new tools. For instance, like Matt Dancy is a scientist in Miami who has developed a machine learning tool called MAVERICK, and this is now accepted for publication in a Nature journal. So this will give it a lot of validity, and I think many more people will use this. Essentially, this software can predict if a certain gene might be an HSP gene, or if certain variants in any gene really could cause disease. And it does this with a really high success rate. In fact, you see here in a sort of model how different similar tools have performed in the past, and on top the two blue lines show you how well MAVERICK performs. So it's a huge step up really in the performance. So what can you do with such a tool? For instance, we can predict if variants in the Spas gene are pathogenic before those variants ever been seen in a human being. So in the small red dots you might see there in the blue square, these are mutations that are known to be pathogenic in the Spas gene. And when the gray dots sort of seem to agree with those red dots, they also predict that these are pathogenic changes. Now, all the gray dots without red, these are purely predicted, and you can see that you could basically have a score ready before a patient is even identified. So when a report comes back and we are not sure if a variant in Spas is pathogenic, we can look at this chart and we can immediately get an opinion from this artificial intelligence tool if this variant is likely pathogenic or likely benign. Here are some other more recent examples of new gene identifications in the broader field of HSP where we were involved. This is a gene called FICD that can carry mutations in HSP patients. This was discovered between the groups of Rebecca Schüle and my own group here in Miami. And we have done some follow-up studies that really of great molecular interest for scientists. And here are some more rather recent examples, and they are certainly not all of the examples we have. And this just shows you what happens when you organize scientists working together. And we think this will continue and even expedite when we establish our HSP consortium. For instance, Rebecca and I have been working under an NIH grant, which has given us sufficient funding and also sort of a great incentive to work closely together. And over the last 10 years, we have published over 60 papers jointly. And when you have a lot of genetic data available and it can be shared and used by scientists, you can then do studies you couldn't do with just a few families or cases in your hand. You can do statistical analysis as shown here. Dana Biss was a student in my lab, and she has done this just a couple of years ago, and trying to use statistical correlation studies in diseases such as spastic paraplegias, but also CMT. We sometimes call them axonopathies. And that also can generate new disease genes and also risk genes or modifier genes for HSP. Now, despite all these many efforts, geneticists have realized that, as I said, we still have this very large diagnostic gap, and that most discoveries we are making only at few families typically. So the success rate or the new genes explain only a small proportion of the missing diagnostic gap. Now, there are certainly more rare genes to be identified, but we also start thinking that we might be missing things by just looking at genes, and we might have to look at outside of genes. And when you look outside of genes in the genome, you really talk about 98% of the entire genome, which is the space outside of genes. And there are certain variations or mutations there in this space that we call, for instance, structural changes or repeat expansion changes. There are examples with diseases where such mutations have been discovered, but not really at scale yet. And a lot has to do with the technology available. And this is changing now with the long-read whole genome sequencing. So we have been especially interested in the last few years in these so-called expansion disorders. And I just want to give you a brief overview what this actually means. There are these repetitive stretches in the genome that we all of us carry. In fact, there is a catalog of more than 1.7 million such regions or loci in the genome. And we have recently studied the largest long-read whole genome data set in the world, about 1,000 such data sets. And we have, just from these 1,000 people, which are not even, don't harbor any disease, we have created over 3.5 billion different alleles over all these different regions. So this just goes to say, this is not just a minor additional place to look at in the genome. This could be a major contributor to disease, and then eventually could lead us to thinking about therapies. And Sarah here is a PhD student in my lab. She also then developed a machine learning tool, again, to fish out from these many different alleles, which ones we should possibly study first, which ones might actually disease causing. So these are really valuable tools. And again, having a consortia, having a unified way of collecting genetic data and clinical data really pushes the development of such tools. And here is one of those examples. Just a few years ago, we and others discovered this gene called RFC1, and there is a non-coding repeat expansion in this gene. And it was a big surprise to learn that this particular repeat expansion or mutation actually explains, is the most important gene to cause what we call sensory neuropathies. So this is a specific disorder where there was a large diagnostic gap, and this non-coding change here now explains about a third of all those patients. And there might be something like this also for HSP to be discovered. And here's another very recent example, really from January 2023, where we jointly discovered this non-coding repeat expansion in a gene called FGF14, which causes ataxia. And in fact, some of these patients also have spasticity. So, and it turns out that this particular repeat expansion now is one of the most common ataxia genes ever discovered. So again, lots of opportunity, and we definitely want to look more into HSP data sets for such repeat expansions. And you already noticed that we work with many different researchers around the world. These papers I showed you have many co-authors. And this is ultimately where we want to go to with our HSP network. And here's just one example what can be done from the ataxia field. And it's kind of unbelievable how many researchers, these are all physicians and scientists from these many different countries, who work together in the ataxia field to solve these very similar problems that we have in the HSP field. And here's another example, the Inherited Neuropathy Consortium also is spanning the world, really. So to sort of close my remarks, it's very important to understand how genetic diagnosis and therapies that target specific genes are really two sides of the same metal. And the immediate goal for geneticists is, of course, to diagnose as many patients as possible and also understand the mutations in those genes that we already know, because not all the mutations or all the variants in, for instance, the spastin gene are necessarily pathogenic. So we also have to understand the individual mutations in those genes, because only those patients that know with a high degree of certainty that their mutation is causing the disease, only those patients will be able to participate in future clinical trials or will qualify for genetic targeted gene targeting therapies, such as gene therapy or CRISPR therapy. And we see it from fields related to HSP that the development of these therapies is at high pace now. In fact, HSP will benefit a lot from the learnings from these fields. I predict that in the next 5 years, we will see multiple really promising gene therapy products that companies will want to test in patients in clinical trials. So we have to basically prepare for this moment, and it is coming. That is for sure. So that's really good news for this community. And I just want to show and thank my colleagues, many colleagues involved in this. And I leave it to Darius to discuss with you, answer questions, discuss the opportunities, and I hope I see you very soon. And I wish you a successful meeting.